Hello, and welcome to DNA Day. My name is Robert Hanner, and I'll be your host and moderator for this discussion. This is the 6th year Canada has celebrated DNA Day. This annual event was developed to help young Canadians explore the world of genetics by connecting them with the science community. It was also created to raise awareness about DNA and the impact genetics has on our world. So why do we celebrate DNA Day in April each year? 2 reasons. First, in April 1953 was when James Watson and Francis Crick published their paper about the discovery of the double helix. And second, in April of 2003, the Human Genome Project was completed. Let's Talk Science and Genome Alberta are the 2 organizations that made today possible. Let's Talk Science is a national education and outreach organization that engages young Canadians in science, technology, engineering, and math through programs designed to make these subjects interesting and relevant for future studies. Genome Alberta is a not-for-profit corporation that initiates, funds, and manages genomics research and partnerships in Alberta. I also want to say a special thank you to Genome Canada, Genome Atlantic, Genome BC, Ontario Genomics, Genome Prairie, the Ontario Science Centre, the Government of Canada, and the University of Guelph for their support on DNA Day. Today, myself and my colleagues from the University of Guelph will discuss DNA barcoding. We will do a quick overview about our research and then answer your genetics questions. If you'd like to submit a question for today's chat, please go to letstalkdna.ca and fill in the form or post a question to Twitter using the hashtag DNADayCDN. Before we get started, I'd like to introduce the panel. As I mentioned, I'm Robert Hanner, and I'll be your moderator. I also have Dr. Dirk Steinke and Dr. Sarah Adamliks, both here at the Biodiversity Institute of Ontario, working with me today. Actually, we've been working together for about a decade now on some pretty exciting stuff that we're thrilled to be talking to you about. So if we can go ahead and progress to the slides. For those of you who can't be here with us in person today, we're at the Biodiversity Institute of Ontario and Centre for Biodiversity Genomics here on the University of Guelph's campus, where we've pioneered DNA barcoding and serve as the global headquarters for the International Barcode of Life project. And I'll be talking about DNA barcodes and food security. But before we get started on that topic, all good stories have to have some actors. And we start with Carl Linnaeus, who brought us the Latin binomial nomenclature for scientific names, giving a genus and species epithet to species about 250 years ago. And so over that period, scientists have been describing species. About 100 years later, of course, it was Charles Darwin who gave us a mechanism for the existence of species through his proposal or hypothesis of descent with modification via natural selection or evolution. So we now know that species exist because they evolved, and we're trying to build a natural classification system for biology. That's the science of taxonomy and systematics, is describing species and their interrelationships. The last 250 years has seen us doing this largely based on the morphology or appearance of adult, typically male specimens. So even though we've described about 1.7 million species over the last 250 years, we can't always apply that knowledge in cases where we need it most, because some species are sexually dimorphic. The males and females don't look anything alike, and we may have described that species on the basis of a single specimen. So we don't know much about its natural history and other forms that it may take, particularly its juvenile stages, where there isn't a lot of morphology to ID them with anyways. And then lastly, when we get into things like processed products, where all the morphology has been stripped away, there's really nothing to try and key out using a dichotomous key that's based on having an adult specimen in hand. But fortunately, DNA-based identification works on all forms. We can sequence DNA of a given species and use that couplet as a lookup table to identify an unknown based on its DNA sequence. And this idea of using DNA to identify species has been around for several decades now. Colleagues here in Canada first published a paper in 1992 on forensically important nucleotide sequences, showing that we could use DNA sequences to identify fragmentary remains. But it was my colleague Paul Hébert here at the University of Guelph who really saw the value in scaling this whole enterprise up to using a DNA-based digital identification system for all of biodiversity. When he published this paper back in 2003 on biological identifications through DNA barcodes, and firmly established this barcoding metaphor in our minds. So briefly, what is DNA barcoding? It's a method of species identification and discovery based on the DNA sequence derived from standard marker genes. And the idea is that for that gene segment, every species will have a unique sequence or a cluster of closely related sequences termed a barcode. And we use different marker genes for different groups of organisms. The mitochondrial cytochrome oxidase 1 gene for animals, some chloroplast genes for plants, an intergenic spacer for fungi, because these genes have been shown to have this desirable property that when we sequence them, we see very little variation within species, but deep divergences between species so that we can use the sequence to tell them apart. And we do this by assembling data in the Barcode of Life data system, or BOLD, which is an online workbench that's hosted here by the University of Guelph to support the global DNA barcoding program. Basically, what we're doing as we're barcoding organisms is we're getting expert identified reference specimens, and we're taking the data on where they were collected in the wild and putting their GPS coordinates in a map, taking high-resolution digital images of those specimens, who collected it, when and where. And then we're also taking a tiny tissue sample. We extract DNA from it, PCR amplify up the barcode marker gene, sequence it, and put that sequence data in along with the specimen data. And that's what forms our reference library. So with that reference library, we're building a master identification key that can help us to identify any life stage. And then we can automate this process so that you don't have to go to a taxonomic expert for a given group of insects or fish and say, can you please tell me what this is? But leverage their collective intelligence through an online information system where we've captured this species and sequence information. And so what do we do with that data? Well, some of the things that we've done with it in my lab are to perform market surveys. One of my roles has been to work with the Fish Barcode of Life campaign so that ichthyologists around the world, that's people who study fishes, for those of you who don't know, have worked with us to send tissue samples here, and we've put barcodes into the database. And so then we've gone out onto the market and collected market samples, generate a barcode for them, compare that to BOLD to get an ID, and then match that with the regulatory framework, in this case here in Canada, to say which species can be sold under a common name. And if that species that the genetics IDs it as is something other than one of the species that can be sold under that common name, we consider it mislabeled. So when we've done this kind of work, we've found widespread seafood substitution across Canada and New York and other parts of the world where we've looked. This came as a bit of a surprise to us because we could put a species level ID on everything that we tested in the market, but 1 out of 4 fillets that we tested was mislabeled. And in fact, 7 out of 9 red snapper samples were mislabeled. So for some of these things, there was a lot of mislabeling happening. And of course, we always see a cheaper species being substituted in for one that's more expensive. So it isn't just simply a case of, I didn't really know what fish I was harvesting. It's pretty clearly economically motivated food fraud. And that's, of course, a real problem for consumer choice because folks like Oceanwise put out seafood buying guides that tell you which species are more ethically harvested or sustainably managed. But if you aren't even able to get the species you think you're buying, we can't use market forces or consumer pressure to change the behavior of exploiting some of these overfished species. So we want to solve some of these problems by bringing more honest labeling to the market. And it isn't just an economic problem. We've seen some cases where people have been poisoned, where they thought they were eating monkfish and it turned out to be pufferfish, in a case in the United States. And it isn't just about food fraud that we're using these tools. There's a whole other angle to this. In terms of food safety and security, we have to think about the systems that we use to produce food. And one of the problems that we have for food production are things like agricultural pests that impact our yields. So we've been building barcode libraries to do more rapid, accurate, and cost-effective identification of agricultural pest insects. And once we've got these barcode libraries, we can then exploit the species-specific patterns of variation in the barcode sequence to make molecular probes that we can take out into the field, so that you can take a portable PCR machine and do a test with a kit right on site to determine if it's a quarantine pest or a cosmopolitan species of no quarantine significance. And that's what's brought us, in collaboration with the Canadian Food Inspection Agency, to help bring these kinds of DNA-based tools to support their regulatory mandate in terms of safeguarding our food and the animals and plants on which we depend.